Welcome everyone, and thank you all for joining today's webinar, How to Prevent Contamination in Cell Culture. My name is Jessica Wagener and I'm a Field Application Specialist for Cell Handling. Here with me is my colleague Daniel Berhan, who is the Global Product Manager for the Cell Culture Consumables.
Hello everybody.
The presentation will take about 45 minutes, followed by a question and answer session. If you have any questions during the presentation, please use the question and answer button on the left hand of your screen. We will take care for your question either at the end of the presentation or we will contact you via email. So let's start now with our webinar, How to Prevent Contamination in Cell Culture. First, we will briefly touch on the types of contaminations, because if you fight something in the lab, you would want to know what you are fighting against. We will also speak about the sources of contamination and some easy ways to prevent it. At the end, we will go through just a couple of tips for aseptic handling. Contamination is an everyday challenge in all cell culture labs. All labs are prone to contamination, but the frequency varies. All labs have contaminations from time to time, but understandable, nobody likes to talk about it. I think we can also agree that contaminations cost time and money. Undetected or ignored contamination actually endangers the reliability and reproducibility of our results. So what do we do against it? In the majority of cell culture labs, 70% ethanol is the #1 substance for disinfecting surfaces, equipment, etc. What we need to keep in mind when using ethanol is that it is not effective against all kinds of contaminants. As you can see in this little cartoon here on the slide, some bacteria die, but others do not, and especially spores are resistant to ethanol. So we know that ethanol is not effective regarding spores, and in addition, it doesn't inactivate all viruses. The second thing most of you and most researchers worldwide use to fight contaminations in cell culture are antibiotics. This used to be a good tool, but it is becoming more and more a poor tool because there is limited protection due to the increasing resistance of bacteria. The most commonly used antibiotics in cell culture are penicillin and streptomycin. Their non-cytotoxic effect was first described in the 1940s. And they are still present in most cell culture labs. As you can imagine, in over 70 years, the continuous usage of these substances supported the increase of resistant bacteria tremendously. So, does it help to simply switch to other antibiotics that are not so common than penicillin and streptomycin? Well, it may be a short-term solution in some cases, but in the end, you are facing the same issue with resistant bacteria. A more general aspect of the routine use of antibiotics in cell culture is that it gets more difficult to detect microbial contamination. Without antibiotics, most of the introduced contaminants would be soon visible under the microscope or even with the naked eye as color changes or turbidity of the culture medium occur. The prophylactic routine use of antibiotics in cell culture not only masks poor aseptic techniques, it can also leave low levels of partially resistant contaminants undetected for longer time periods and therefore creating a greater risk for the whole lab or facility. Besides the effects of antibiotics on bacteria, what most people ignore is that it is known that antibiotics can have side effects on the metabolism of cells. Whereas the usage of standardized culture media and their supplements is widely established and accepted, the prophylactic usage of antibiotics varies tremendously. Different researchers use different types and concentration of antibiotics for the same cell types. Some research don't add any antibiotics to their cell cultures. So, in most cases, the data that are published for a certain cell type were generated under non-standardized conditions. This, of course, can be problematic when it comes to reproducibility and reliability of experimental data. So taken together, antibiotics are not really good in protecting you from bacterial contaminations, and they are not really good for your cells. This is why we would like to show with this webinar that there are simple and efficient ways that offer huge potential to protect your cell culture from contamination. So before we start with the next chapter, we would like to ask you to participate in our first poll.
So the first question is regarding contamination. Did you ever have problems with contamination in your cell culture lab? And the answers could be never, very rarely, or on a regular basis. So please click on the answers and vote now, please. Okay, so we're just waiting. Answers coming in now. A few seconds more. Okay, now we'll stop the poll. Thank you very much, and publish the results. Okay. Thank you very much.
Okay, so thank you for your participation. So we see that over 70% of you experience contamination very rarely, and 20% see contaminations of their cell culture on a regular basis. So let's start with the second topic. Let's see who our enemy is. In general, we find two major types of contamination, the microbial ones and the eukaryotic. The microbial contaminations are fungi, yeast, bacteria, and mycoplasma. The other ones, the eukaryotic, are not necessarily so well known and are cross-contamination of other eukaryotic cells and cell lines. Now we want to look at all of these in more detail. Let's start with the more common types of contamination, fungi, yeast, and bacteria. In most labs, it is one of these three types if there is from a study where companies and institutes asked research labs to send them aliquots of the cell lines they are working with, of course high-quality stocks. So research labs sent their stocks to the institutions, and what came out was that of all the labs sending out their good quality stocks, actually 6% had contamination. If you ask me now how it would be possible that we have such a high number of contaminated stocks that go undetected, when usually we can easily see them in the microscope, I can tell you that the reason is probably due to antibiotics. In this case, it might be the inadequate use of antibiotics in the cell culture with low concentrations, so low that they actually don't kill the bacteria anymore. These inadequate concentrations, however, are not miscalculations, but result from repeating warming up of the complete medium flask, including antibiotics. Most antibiotics are temperature sensitive, so the repeated warming lowers their amount in the culture medium. This is probably the reason why in some labs those undetected contaminations can occur. The next and not so nice contaminations are special types of bacteria called mycoplasma. They are very small, have no cell wall, and grow as parasites attached to the cell membrane. The bad thing about these guys is that they are not detectable in light microscopes. So even if we monitor our cell lines regularly, we wouldn't detect them. They don't flow in the medium like other bacteria, but are attached to the cells. That makes them somewhat invisible. In order to find whether your culture is clean or contaminated, you need to check the culture, for example, with a PCR test. Why are mycoplasmas not good for our cells? They secrete toxins that are not good for the cells. The other point is they grow very fast, actually faster than your cells, and therefore competing the nutrition in the culture. So your cells might actually starve due to mycoplasma contamination. And this is another reason why a latent contamination goes undetected. If you are on a good feeding schedule, your cells actually might not suffer, although there is a persistent mycoplasma contamination. I want to go a little bit deeper into the topic of how mycoplasma affects cellular metabolism by showing you some data from a recent study published last year. These researchers infected HeLa cells with Mycoplasma and looked at the differential gene regulation in the cells up on different time points of infection. As you can see in this graph, the number of genes that respond to the Mycoplasma infection increases dramatically with time. And I just want to briefly show you an extract of the list of genes that were identified. And I know it is hard to read, but let me tell you that the cellular effect was dramatic. The genes that were deregulated upon infection are associated with signal transduction pathways, immune responses, cell cycle regulation, cell growth, and other cellular functions. So I think that we can all agree that Mycoplasma contamination can have dramatic effects on cell cultures. Now I want to switch to a complete another type of contamination. Cross-contamination means that a given cell line is contaminated with another cell line. How can that happen? Usually this happens by an accident somewhere during the handling of cells. So it is a mistake in aseptic handling. For example, you are supposed to trypsinize one cell line after the other, but since you are a little bit in a hurry, you handle another more aggressive cell line here in pink at the same time. By accident, a little droplet of the more aggressive cell line gets into the blue culture. Because the pink cells grow faster than the blue ones, with the following steps of cell culture, the pink cells become more numerous and start to overgrow the blue culture. After a given number of subcultivations, you will end up with a dish of pink cells or a mixture of both. So, for example, if the blue cells would be Vero cells and the pink ones would be HeLa cells, you would end up with a dish that is labeled Vero but actually contains HeLa cells or a mixture. As soon as two cell types are mixed up, it is very hard to distinguish those two. This process goes fast and might go undetected, especially if the morphology is not drastically different. We know this problem already since 1967, when the first publication came out. Still to date, we know that 15 to 30% of all cell lines used are either cross-contaminated or misidentified. Currently, there are more than 400 cell lines identified to be cross-contaminated. So what all of those types of contaminations have in common are their consequences. Loss of time since you have to redo the experiment, increase in cost, time is money and more material. Loss of reproducibility, you might have completely different responses when you work with contaminated or non-contaminated cells. The last point might be the most horrific, the loss of significance. If you want to find a new therapeutic treatment of breast cancer, but you work with melanoma cells, your work is useless. To summarize, avoiding contamination is much easier than coping with it. In order to efficiently fight the contamination, we need to know where it comes from. And this is what we look at in our next chapter, sources of contamination. We distinguish between the indirect and the direct contaminations. Indirect contaminations come from the labs or the equipment or even from ourselves. Direct contaminations come from reagents that might be contaminated, or you use cells that have been contaminated before you took them in culture. So we start again with labs and equipment. You might have airborne germs that come from ventilation and opening the doors, hopefully not from opening the windows, which should be prohibited in cell culture labs. Then we have the water that might bring in germs. This might be water from the tap, the water bath, or the water tray inside the incubator. The water might carry two different types of germs. First, the primary germs that are brought with the water in the lab, but also you have secondary germs that were originally airborne germs. For instance, in your incubator tub or your water bath, you would probably use distilled water or autoclave it before use, so it is free of waterborne germs, but the water is in contact with the air and germs might literally fall into the water with the air. The next source for contamination is dust and dirt, which is a nutritional base for germs. So the more dust you have, the more ground you have for potential contamination. The next indirect source can be the personnel. We all carry bacteria. They belong to our flora of mouth and nose, and they can affect the cell culture. When we are ill, we even carry more germs and in addition viruses. The clothes we wear and wear underneath the lab coat are the clothes we are wearing on our way to work, and they have been in contact with the outside air and other sources of contamination. So some microbes might stick to the clothes and we carry them into the lab. Same is true for the hands and other skin parts. Apples, for example, can have large amounts of yeast on the surface, which is not bad if we eat them. But if we don't clean our hands before entering the cell culture lab and starting to work, this might affect our cell culture. Our hands can also carry mycoplasmas. This is not where they normally grow, but if we touch our mouth or scratch our face, they might stick to the hands. The shoes are also a major source of contamination. They carry traces of soil and dirt, and we carry the microbes that are in there in our cell culture lab if wearing street shoes there. Let's move on to the direct source, the reagents. We use a broad range of reagents in cell culture. Media usually are rarely delivered contaminated nowadays. They are sterile filtered, and it happens very randomly that you receive them contaminated. FBS is a different story though. It used to be a very important source for contamination, especially for mycoplasmas. This situation has greatly changed since most production facilities usually filtrate their FBS now at 0.1 microns, which filters mycoplasma more efficiently, just not as efficient as other bacteria due to their small size. Also, they don't have a cell wall and are therefore quite flexible. In addition, there is also gamma-irradiated FBS out there, which further increases the safety of the products. Production facilities and labs working under GMP/GLP conditions do routine testing on the FBS that is used. Something that reversely changed over the last 10 to 15 years is that we now use reagents that are produced in cell culture. Before that, these reagents mostly came from bacterial sources. Antibodies or peptides you receive from cell culture might be contaminated if the production culture was contaminated. And then, any aliquot prepared in the lab carries a potential risk of contamination. The person preparing the aliquots might have made a mistake in aseptic handling, and your stock might carry a contamination. Before I go to the next slide, we have another poll and ask you to participate by clicking.
So the next question is regarding cell types. What kind of cell types do you use in your lab? So the answers could be cell lines, primary cells, or both. So please vote now. The answer is coming in. Thank you very much. So we're waiting just a few more seconds. Okay, now we close it. Okay. Push the results to you.
Okay, so we see that almost half of you is using cell lines, and also about 45% is using those cell lines and primary cells. Okay, so thank you for participating. Let's first talk about the risk when working with cell lines. If you introduce a new cell line into the lab, you have a potential risk of introducing, you have a potential risk of introducing a contamination to the lab. Also, you might rely on older stocks that simply never have been tested. If you have never tested old cell lines, please test them immediately to make sure that they are clean. Remember the numbers we have been seeing before? 6% of the cell line stocks that thought to be clean were contaminated by different microbes. The second contamination, as mentioned before, might be a cross-contamination. One number we learned about was 15 to 30% of all cell lines used are cross-contaminated or misidentified. For primary cells, the situation is a little bit different because they are finite. They die off at some time point, and you have to generate or purchase new cells. Primary cells mostly are contaminated due to the semi-sterile tissue preparation. There is no possibility to sterilize tissue samples, and in most cases you just place it in a sterile container. Upon cultivation, you have to make sure that you don't get microbes into your freshly prepared tissue. So your primary cell cultures always carry the risk of showing microbial contamination. With primary cells, you have hardly the risk of cross-contamination because they have a limited lifespan. In case your primary cells suddenly become immortal, you should think about cross-contamination. Spontaneous immortalization is hardly seen in cell culture. Of course, you should validate that you isolated the cells of interest. So, for instance, make sure that you isolated smooth muscle cells and not fibroblasts. Again, cell authentication is an important factor and should be considered as a routine step in cell culture. I want to end this chapter by showing you two sources that I think can be very helpful for you in your daily cell culture work. The first one is a recently updated guideline for good cell culture practice that was published this year in the British Journal of Cancer. These guidelines cover all relevant areas such as cell authentications, cryopreservation, transfer of cell lines between labs, and microbial contamination, and more. The second source I want to point out to you is the website of the International Cell Line Authentication Committee, or ICLAC. You can download a list of cross-contaminated and misidentified cell lines that is constantly updated and monitored by the committee. To look into this database is the simplest way of checking the cell lines you are currently working with or the cell lines that you are proposed to work with. On this website, you can also find advice on how to incorporate cell authentication into your everyday cell culture practice. But before you start reading, let's come to our next chapter. So now we know who our enemy is and where it comes from, so the next logical step is preventive hygiene. Before we start the chapter, we would like to start the next poll.
So the next poll question is regarding cell culture techniques. How did you learn cell culture techniques? Learning by doing. My colleague in the lab taught me how to work with cells. I attended a training course, or I attended lectures. So please vote now. So answers are coming in now. Thank you. Just wait another second. Okay. We close and show the results to you.
So we see that the majority of you learned how to work in cell culture by their colleagues. So knowledge of cell culture is given from person to person. So we want to talk a little bit of preventive hygiene. From this slide, you may recognize that it looks like the first slide of the previous chapter. We have indirect measures of preventive hygiene and direct measures. We divided the chapter in this way because we would like to convince you that you should take precaution individually for each type of potential source of contamination. There is not the one way to wipe out the risk of contamination. So for every source, choose a special approach for preventive hygiene. Before we look at what we do, we would like to introduce you to a general idea of preventing any unnecessary transfer of germs into the lab. That sounds very easy, but most labs don't actually do that. So here's the idea. If you work in a cell culture lab, you work under a biological safety cabinet, or BSC, like depicted in the scheme. Within the biological safety cabinet, we have a sterile atmosphere that we rely on, so the BSC allows us to perform our work in a sterile way unless we carry germs into the BSC. There is a potential risk that germs get transferred into the BSC, and we would like to convince you that a reduced germ load in the cell culture lab reduces the risk of carrying germs into the BSC. Outside the germ-reduced cell culture lab, you can have your normal facility without regulations and without preventive hygiene. But at the door to the cell culture lab, take some precautions to reduce the germ load. This is what some institutes don't do. They do treat that cell culture lab a lot of times like a regular lab. So the blue area is also green in these facilities. Before you enter the cell culture lab, wash your hands, disinfect, and use hand cream. Healthy skin has less germs than dry skin. When you enter the lab, put on gloves immediately and disinfect them. Don't walk into the lab with street shoes. Put a rack with lab shoes next to the cell culture lab door, and don't walk with these shoes everywhere. Put them on only before entering and put them off when you are leaving the cell culture lab. Inside the cell culture lab, a separate air filtration system and a positive air pressure can help to keep airborne germs out when the lab door is opened. To limit the source of contamination, only people who actually have to work in the cell culture lab should be allowed to enter. Personal protective equipment is a must in cell culture. It is not only to protect yourself, but it is more to protect the cells. The lab coats, gloves, and shoes you are wearing inside the lab should be restricted to the cell culture lab, and if possible, use a gowning room or area close to the lab as a sluice where you put on your cell culture equipment. It is common that the complete lab is cleaned once a month, but of course this is depending on the lab. How big is it and how many people are working there? In the next few slides, I want to talk about the typical equipment that is found in every cell culture lab. Let's start with CO2 incubators. Due to the door openings, the inside of the incubator is never sterile. Every time the door is opened, germs can enter the incubator, plus the atmosphere inside the incubator is disturbed. Therefore, door openings should be reduced in duration and frequency as much as possible. If you have an incubator with a split inner glass door, you can further decrease the contamination risk and disturbance of the atmosphere. A standard cleaning routine should be part of every cell culture lab. The humid atmosphere and warm temperature inside the incubator are not only necessary for the growth of your cells, but also support the growth of contaminants. Therefore, you should take special care about cleaning and disinfecting the incubator on a regular basis. If you have an incubator with a seamless, weld-free lining, you can simply clean the inside by wiping it. If the incubator chamber has corners, fissures, or seams, you should pay special attention to these areas and clean them, for example, with a brush. Run the automatic disinfection routine if it is available for your incubator, at least once a month. An incubator with copper interior also reduces the risk of contamination. The copper lining reduces bacterial growth. The water tray inside the incubator is necessary for keeping the atmosphere humid. That avoids the drying out of the cultures. The water inside the tray should be autoclaved and exchanged regularly, because it is a source for contamination, as airborne germs can grow in the water. If the incubator has a fan inside, the germs get distributed in addition. Besides changing the water inside the tray, regularly you should supplement it with additives that are reducing germ growth. If you have an incubator that heats and vaporizes water, though, don't use additives, because they might affect your cells. Let's come to another standard device in the cell culture lab, the water bath. It is used for warming media and reagents and for quick thawing of frozen cell stocks. Due to the temperature and the humidity, the water bath is an important source of contamination. So it should be cleaned thoroughly on a regular basis. Besides that, the addition of bactericides and fungicides to the water is recommended. When you remove the vessels from the water bath, make sure that you disinfect them properly before placing them in the BSC. Pay special attention to the cap area when disinfecting, because this is the place where germs can hide. Talking about pipettes. All pipettes or pipetting aids should be restricted to the cell culture lab. A good advice is to simply put a sticker on each pipette and label it with cell culture, so everyone knows that this pipette should be kept inside the cell culture lab. Disinfect the pipettes each time before you place them in the BSC. In addition, you can disassemble the pipettes to clean it from the inside. For keeping precision, pipettes should be calibrated at least once a year. In general, all devices and surfaces in a cell culture lab should be wiped and dusted regularly. Wherever possible, use dust coats, for example for microscopes. Spills should be cleaned immediately, and if available, use centrifuge equipment that is autoclavable. Despite the right handling and taking care of equipment, there are some do's and don'ts that you should keep in mind when entering a cell culture lab. Always wash your hands before entering and when leaving the lab. As mentioned before, you should have personal protective equipment that is restricted to the cell culture lab. It will help to keep germs under control that are on your clothes, shoes, or skin. Wearing a mask might sound a bit paranoid to you, but it can be a good idea in case you have, for example, a cold or another kind of infection. Let's come to the direct sources of contamination and the measures that can help to avoid contamination. Every aliquot of FBS, medium, or other reagents should be labeled with a lot number in order to be traceable. Risky materials like FBS should be subjected to an intake quality control, like you see here in this scheme. Only when you are sure that the new batch of FBS that you purchased is free of contaminants, you can start working with it in your regular cell culture. As mentioned before, mycoplasma are a special threat in cell culture labs because they cannot be seen with a light microscope. In addition to the intake quality control I showed you on the previous slide, a test for mycoplasmas can reduce the risk of direct contamination through reagents. There are a number of ready-to-use PCR kits on the market for testing of different types of mycoplasmas. After the cultivation, the whole analysis via PCR only takes you about 1.5 to 2 hours. Another direct source of contamination is the cells themselves. When you purchase new cell lines or primary cells, make sure that you get them from a reliable source, like the American Type Culture Collection or the European Collection of Cell Cultures. When cells are given from lab to lab, or when you isolate primary cells from tissue, these cells should be considered as potentially contaminated and be separated from the other cells until proven absence of contaminants. Reserve one shelf in the incubator for new cells, or, if possible, use a separate incubator for true quarantine. In general, it is a good advice to always keep primary cells and cell lines separate. We already talked about cross-contamination of cell lines and the importance of cell authentication. So here comes our next Paul.
So the next question is regarding exactly the cross-contamination of cells. Have you ever encountered cross-contamination with a different cell line? So never, very rarely, or several times. So please vote now. We're just waiting another second. Okay, thank you very much. Now we will close the poll and show the results to you.
Okay, so we see that the majority of you never experienced cross-contamination, but almost 30% already experienced cross-contamination with a different cell line. So let's see how we can test ourselves for cross-contamination. Cell line identity testing using short tandem repeat profiling ensures you that the cells you are working with are the ones that you think they are. To be completely sure that the cell profile has not changed over the course of a study as a result of cross-contamination, you might consider STR analysis at the start, middle, and at the end of a project. You should consider regular checking of STR profiles, especially for long-term studies with one cell line. In our last chapter, we want to give you some simple tips for aseptic handling. As for all other equipment in the cell culture lab, the biological safety cabinet should be cleaned on a regular basis. As this is the area where you open the dishes, plates, or flasks with cells, there are some things to be kept in mind to avoid contamination. Disinfect the working area in the BSC completely and let it dry before every use. Disinfect every piece of equipment that you bring into the BSC. Every item that is in there disturbs the laminar airflow that keeps germs outside. So don't overcrowd your working space with items that you don't need for the particular working step. When working at the BSC, it is a good advice to have one clean site with culture media, pipettes, and new consumables, The other side is reserved for liquid waste, used tips, and so on. Again, any droplets or spills need to be cleaned immediately. Your hands are the major source of contamination when handling cells. Disinfect the gloves sufficiently. That means let them dry before you start working. And disinfect them not only once, but repeatedly. For example, when you adjust your glasses, never do it without subsequent hand disinfection. When you are pipetting, try to avoid moving your hands or arms over open vessels. In general, avoid hectic movements with your arms inside the BSC and when standing or sitting in front of the BSC, as it will disturb the laminar airflow. To minimize the risk of cross-contamination, only work with one cell type at a time and prepare separate medium bottles or aliquot for each cell type. Before you start working with the next cell type, disinfect your working space inside the BSC. Just a few words about pipetting. When you need a precise volume, hold the pipette vertical when aspirating liquid. Pipette slowly to avoid the risk of contaminating the pipette itself. When you are releasing the liquid, you can tilt your hand to avoid that your hand is directly over an open vessel. Pipette with a medium speed to avoid any droplets. Using filter tips protects the cells from being contaminated, and it protects the pipette from being contaminated. So you might consider using filter tips at least for the very important steps like freezing of cells to build up stock cultures. In the last 3 slides of my presentation, I want to talk to you about the vessels that are in constant and in direct contact with your cells, the cell culture consumables. When you open a plate, dish, or flask, the lid should be placed outside the working space, but inside the BSC, of course. Some people hold the lids in hand while pipetting, but this requires practice and routine because you have to avoid touching the inside of the lid while holding it. When you are placing the lid aside, you can position it inside up or inside down. Both ways are okay when performed correctly. That means when you put it inside down, make sure that the surface is clean, and when you put it inside up, make sure that you don't move over it with your hands or arms. When you use Eppendorf consumables, I recommend to put the flask cap onto its side because thus it is not in contact with the work surface nor the airflow. Its anti-rolling design will make it stay in position. The Eppendorf plate lids have little support hubs that minimize the surface contact when the lid is placed inside down. The routine steps in your daily cell culture work include a lot of transporting the cells from A to B and back again. I give you an example. You take the cells out of the incubator, put them under the microscope to check if they are ready for splitting. You carry them to the biosafety cabinet, exchange the medium or split them, and then you bring the cells back to the incubator. Sometimes, for example, when you're trypsinizing the cells, you go again from the incubator to the microscope, to the biosafety cabinet, and back to the incubator. Transporting flasks, dishes, or plates with medium inside always carries the risk of spilling. When using flasks, make sure that you carry them slightly tilted upwards to avoid medium contact. In addition, flasks with a hydrophobic filter give you extra safety. Dishes with a corrugated gripping ring and plates with a gripping area give you the safety when transporting them. In addition, the Eppendorf dishes have a special ring inside their lid that traps spills inside the dish. When you want to take one or a few dishes from a stack of dishes in the incubator, the gripping ring avoids unintended opening of the lids, and so does the gripping areas of the plates. I'm coming now to the last slide of the presentation, and that may be the simplest advice I can give you to avoid cross-contamination. Clear labeling of cell culture vessels with an ethanol-resistant pen. With that simple measure, you can prevent accidental mix-up or misidentification of cells. Every cell-containing vessel, no matter if it is a dish, a tea flask, a multi-well plate, or a tube, should be clearly labeled with the cell name, passage number, date, and lot number if applicable. So this is the end of the presentation. Thank you for your attention so far. And Daniel and I will need just a few minutes to go through your question, and then we will start our question and answer session. In between, we will show you a little video about the new Eppendorf cell culture consumables, and we will come back to you in a few minutes. Okay, so I hope you could all see our video. I'm not sure. I hope so. So we had a few questions. Okay, so I think we are back on air now. Okay, thank you. I hope you could all see our video. So we would start now the questions and answer session. There was quite a few questions coming in. If we are running out of time or if it's more complex question, we will follow up via email. So be sure that every of your question is answered.
Okay. So thank you very much for all the questions. We got really a lot of questions. Thank you very much. So one of the most asked questions was, can we get this presentation sent to us as a PDF? Jessica.
Yeah, of course. We will provide the presentation as PDF.
Action.
Okay, I hope you can hear us now. I'm just checking with Tracy.
Okay.
Okay, so we leave on the phone now. Okay, we have a little technical problems here. I'm so sorry.
Okay, I hope that you can hear us now. So as we are not sure if you could hear the first question, we just repeat it. So the first, one of the most answered questions was, can we get this presentation as a PDF or as a summary sent to us via email?
Yes, we will provide the presentation as a PDF file for you. So yes to that.
Okay. So a lot of people were interested in the mycoplasma testing. So what is the best way to do it? How often should I do it? And does freezing of my samples actually affect the mycoplasma detection?
Okay, so maybe first an answer to the last part of the question. The freezing should not affect the mycoplasma test. And the question how often you should do it really depends on the frequency. I mean, if you're working in a lab where a lot of new cell lines are used, or a lot of different cell lines, or primary cells and continuous cell lines, a mycoplasma test on a regular basis is recommended. So, but it really depends on the lab. So if you do it once a month with every cell type that is currently inside the incubator, it should be fine, but to do it more often is not wrong here. So make sure that all your cell stocks are mycoplasma tested, so all of the cells that you have in the liquid nitrogen. If you haven't tested them before, make sure that these are tested. And then do it with the cells that you have currently in culture on a regular basis. So I would recommend here at least once a month.
Okay, then another question was regarding the FBS. Is heat inactivated better?
Yes, heat inactivation is better than non-heat inactivation, but you want to make sure that it definitely is filtered and not only heat inactivated.
Okay, there were also some questions regarding alternatives to water baths. Are there any alternatives? I mean, of course, you can heat your samples in a different way.
Well, some people use the incubator for heating or warming aliquots of their media. This, of course, is also a risk of contamination because you have it in the incubator, but it is not an additional water source like you have with the water bath. So that might be an alternative to that. You could also use heating blocks for tubes. So that might be a solution as well.
Okay, so we have a lot of questions that are coming in right now, actually. So regarding different cleaning routines and so on, I would say that because they're very specific, that we will of course contact you and answer that questions via email after the presentation. Again, thank you very much for your attention. From my side.
Also from my side, thank you very much for taking the time for joining this webinar. We hope we could give you some helpful advice, and we will follow up your questions that are coming in right now via email within the next days. Okay, so thank you again for your attention and have a nice day.
Goodbye.
Bye-bye.